All right, so a lot to get through today. There's a lot of people here that are either junior students or faculty who do not know much about immunology, so I'm going to try to start from the beginning and go through things. But again, interrupt me if you have any questions. All right, so whenever I start talking about immunology, I like to teach that the immune system can be thought of best as operating on two axes. The first is the self and non-self axis, and the other is the danger versus safety axis. And the idea is that if your body recognizes something as non-self in an environment of danger, that's when you're going to get an immune response. And if you don't have both of those, you're going to get some sort of tolerance. So basically, you're only going to make an immune response when your body recognizes that there's something non-self or foreign in an environment of danger. When we think about the immune system, it is an incredibly complicated biological system. Oftentimes, it's thought of in terms of the different cells that make up the immune system. So you can see here there are myriad different cells that make up the immune system. They can, in general, be classified into one of two categories. The first is the innate immune cells. And so these are going to be things like macrophages, basophils, and these are going to be the things that will instantly be able to recognize and respond to a foreign pathogen, for example. Neutrophils, monocytes, macrophages, NK cells are another one of them. And so innate immunity is thought of as being rapid. So it can very quickly respond to a pathogen, but it's also non-specific. It can't learn which pathogen you have been exposed to before and can't do anything different the next time. It's also constant, right? So basically your innate immune system is constant throughout your life, more or less. The other big part of the immune system is what's known as the adaptive immune system. And the cells that are responsible for that are two main types of cells, T cells and B cells. And adaptive immunity is characterized by a delayed response. This is the reason why if you get sick with COVID or something like that, at first, everyone was very susceptible to COVID because no one in the human population had ever experienced COVID before. And it took your adaptive immune system a while to recognize and respond to it. But it's also specific. So once you have been infected with that specific COVID virus, you shouldn't be able to be infected with it again because you have this very specific adaptive immune response that will protect you from it. And lastly, that leads to what's known as lasting memory. So once you have an immune system, immune response, an adaptive immune response to something, you'll have memory for the rest of your life if you ever see that same pathogen again. All right, so HLA really has more to do with the adaptive immune response. And so we'll talk a little bit more about the adaptive cells. And as I mentioned, these are going to be two types of cells. You have your B cells, and their main function of B cells is to produce antibodies. Antibodies are often shown as these Y shapes, and the idea is that these antibodies have very specific receptors that can bind to antigens and either neutralize it or induce some sort of function. And they can neutralize microorganisms, they can coat and kill cells, these antibodies. The other big type of cells for adaptive immune response are the T cells. And the T cells some of them are called killer cells, and they can actually kill an infected cell, right? So if a T cell recognizes that a cell is infected with COVID, for example, then it can kill that infected cell. T cells can also help other cells. For example, they can help B cells make antibodies, or they can help macrophages kill bacteria that macrophages have engulfed. And then lastly, T cells can also regulate the immune response. They can, there's T cells known as regulatory T cells that actually suppress immune responses. All right, so I talked a little bit about adaptive immunity and how it has a delayed response. This is a cartoon showing, for example, if you are exposed to antigen A, and we can think of this, for example, as being a specific COVID strain. If you're exposed to it, it will take a while before you get this adaptive immune response to this antigen A, and then that will slowly go down after you've cleared the infection. And when you have a secondary response, meaning if you ever see that same pathogen again, you will have a very quick secondary response. And so this is what would be that memory response. However, if you see a different pathogen, let's say you see a different COVID virus, then you will have a brand new response to that, and that's what would be antigen B, right? So that's the whole idea of a specific response. It's only specific for one pathogen, but not another. And then you have this lasting memory that can come back up very quickly to protect you so you don't get the same infection again. Another thing to think about when we're talking about adaptive immunity is it happens with what's known as clonality. And the idea here is I'm showing lots of different B cells. Each of these B cells has a different antibody receptor on them. And what I'm showing here is if you're first exposed to a pathogen, for example, only a very small subset of all of your B cells will be specific for that specific virus. But after you have been exposed to it, these cells will clonally expand. So this one cell here will expand into many clones, many copies of that same cell. And so now at the second exposure, you have significantly more of these cells that are specific for that virus. And so that is one of the ways that memory response can happen, because you have an expansion of these clones. So I've used a SARS-CoV-2 COVID as an example for some of these, but I'll kind of go through and walk through how this would work. So if you have an infection, the virus would invade lung epithelial cells. They would start replicating inside of these cells. They would kill a bunch of lung cells, and this would induce lots of inflammation. And so because of that, this is going to be really on this non-self, right? Because SARS-CoV-2 is definitely a non-self protein. It's not something our bodies make. And because it's killing so many cells, it's also going to be on this danger axis. And so this is, rife for the type of immune response that's going to cause an immune response. So let's see the way this would work. You have these epithelial cells that are infected by COVID. These would send signals out, would call in different sorts of immune cells into there. And then these immune cells, I'm showing here, this one cell that's specific for SARS-CoV-2, will then become clonally activated and will make more of these clones. And then these, I'm showing here a B cell, which will turn into a plasma cell, which is a type of B cell that makes antibodies. And then those will be clonally expanded. And so now you have lots of these cells that make these antibodies specific. And so all of these will make soluble antibodies. So now you have antibodies coursing all throughout your blood. And then these antibodies, let's see, I'm showing here, T cells will come and directly kill these infected cells. And then you'll have the antibodies, which will come and neutralize all of these SARS-CoV-2 viruses that are outside of your cells. And so this is really the way that you have your adaptive immune system, both the T cells and the B cells working together to kind of fight this infection. So the whole thing I've been talking about an infection at this point, right? And so that's really the best way to think about it, because the immune system really evolved in response to infection. But what about transplantation, right? We're all here because we're working on HLA in the guise of transplantation. All right, so the whole idea with transplantation is, so I'm showing here a heart. Our project deals more with kidney transplant, but it's very similar ideas. So the whole idea here is if we have a heart transplant, we want to try as much as possible to get the heart to be viewed by the body on more of a self way and more on the axis of safety, right? So if we put an allograft in, if we put a transplanted kidney or a heart in, we want the body to see it as safe and to see it as self as much as possible. And so when we're thinking about HLA and we're thinking about adaptive immunity, this is really going to be more on the self versus non-self axis. And so the idea there is when we're thinking about how HLA works, we're really thinking about this one axis of self versus non-self. And the way this works, the way HLA gets involved, so I wanted to show a little bit about how the immune system develops. And so I'll start here with B cells and how they figure out self versus non-self recognition. So B cells are named because they come out of the bone marrow, B for bone marrow. So what happens is you have a bone here and you'd have B cell precursor come out of the bone marrow. They would then start making B cell receptors or antibodies, another name for antibody. And so each one of these cells would make a different specific antibody. And then these antibodies would be negatively selected for any antibody that would recognize a self protein. The whole idea is your immune system is trying not to have any cells that would react with self proteins. It only wants to have cells that would react with non-self proteins. So during development here, any of these antibodies that recognize self proteins will be negatively selected or killed off. And so the only thing that will remain then is this mature B cell that does not interact with the self protein. So, when we think now about mature B cells, which is what's going to be out ready to participate in an immune response, these are going to be cells that were negatively selected against self. And so they're only going to be specific for non-self specificities, and they're not going to respond to self antigens. And so that's really the way that a B cell can determine if things are self versus non-self.
I was just curious with the early determination of self. Could you explain? So does this happen in the bone marrow? Where the cells are generated, or so this testing to make sure that it's self versus non-self?
Yes, right. So B cell development all happens in the bone marrow, right? And so it's interacting with self proteins that are normally expressed during just kind of steady state response, steady state when you're alive.
Gotcha. And like, what does it use as the array of proteins or potential antigens to give examples of self?
That is a great question, Ryan. I will say that I don't know as much about B cells. I know a lot more about how it works with T cells, and I can discuss it in a couple of minutes here. I will say that there is a lot more mechanisms for the T cell response, and part of that is because in general, B cell responses can't work without T cells anyway. So there's more selective pressure to make sure that the T cells are kind of the master regulators of immune response. And so even if you have some B cells that can recognize self, they're never going to get very far because there won't be a T cell that will help them and say, yeah, go ahead, this is a real immune response. All right, so back to T cells. So the big difference between T cells and B cells have these antibodies. And so they're proteins that are either stuck on the surface of a B cell or they're secreted and they are part of the plasma in your blood. But T cells, they only recognize something by what's called a T cell receptor that is always attached to the T cell. So there's nothing that a T cell secretes in order to have its function. And a T cell always interacts with something in an HLA molecule. So now we're finally talking about HLA. So the whole idea is that T cells, in order to determine what they're going to respond to, they have to interact with an HLA molecule. All right, so T cells recognize the HLA molecule and specifically a peptide antigen that's presented by an HLA molecule. I'll go through this a little bit more, but the idea here is, you have this T cell, the T cell receptor, and to determine what it's going to respond to, it interacts with this HLA molecule here and this little peptide antigen that's presented within it. All right, so I need to talk a little bit more about that. So in here, I'm zooming in on the T cell receptor here at the top, and the HLA molecule here at the bottom. So the T cell receptor is present on the extracellular surface of a T cell. An HLA molecule is present on the extracellular surface of these specialized immune cells called antigen-presenting cells. These are going to be things like macrophages and dendritic cells that have these HLA molecules on them. And the whole idea is that we have a whole bunch of different HLA molecules, and they can all present these peptides. So here, if we take this HLA molecule over here and we kind of turn it on its side, now we're looking straight down on the top of an HLA molecule. And what you can see is this blue protein structure and this green protein structure are two parts of the HLA molecule. And right in the middle is what's known as this peptide binding groove. And so you can think of it a lot like two hands that are here that are able to hold on to a piece of a protein. A piece of peptide is a small piece of a larger protein. What I'm showing here is a bunch of amino acids that came from this larger peptide, this larger piece of a protein. And this is essentially what a T cell is going to see when it determines if something is self or non-self. Right, and so we can think about this. Let's go back to the COVID infection. Right, so if you're infected with a COVID virus, well, the COVID virus is going to have a bunch of proteins that are specific to COVID. Some of those will get chopped up and they will be presented in this HLA molecule, and they'll be presented to T cells. And now when a T cell comes along, it'll interact with the HLA molecule, and it will look at that peptide that's being presented there to determine if that is self or non-self, and if it's going to make an immune response. All right, so I'll talk a little bit more about how this works. So I talked about how T cells recognize HLA in a peptide in this complex here. T cells are named T cells because they come from the thymus. And so the idea here is that you have these T cell precursors that come into the thymus. And then much like a B cell, they rearrange and make these T cell receptors, right? So now you have these T cells that each one of them has a different T cell receptor on it. And they go through a couple of different types of selection. So when I was talking about B cells, they only had one type of selection where if they interacted with anything that was self, they were just deleted. T cells first go through what's called positive selection. And the idea there is that each one of these T cell receptors has to see if it can interact with an HLA molecule at all. If it can't react with any HLA molecules, it does not pass positive selection. So what I'm showing here is this particular T cell receptor was not able to interact with an HLA molecule, so it was killed off. And the only thing that's left now are these two T cell receptors that can interact with an HLA molecule. The next thing that happens is what's known as negative selection, where now it starts looking more at the peptide that's in there. And if any of these T cell receptors react too strongly with this HLA molecule and that peptide, they will be negatively selected. And so what that means here is that this yellow one here, it reacted with the HLA molecule, but it reacted too strongly with that self peptide, because if we think about in a normal thymus, as T cells are being educated, they're only going to be exposed to self peptides. And so what happened here is this yellow T cell receptor interacted with a self peptide too strongly, and so it was negatively selected. And so the only thing that's left are these mature T cells that can interact with an HLA molecule a little bit, but not too much. It's kind of like this Goldilocks zone. And so the idea here is that you have these T cells with these T cell receptors that you know can interact with your HLA molecules. And if there ever comes along an HLA molecule that's presenting a foreign peptide, then one of these T cell receptors is probably going to be able to recognize it really tightly and start an immune response. And so, Ryan, to answer your question, there is a really cool set of machinery within the thymus that specifically expresses a bunch of tissue-specific self-peptides that would not normally be expressed in the thymus. And it's this master regulator called AIRE, A-I-R-E. And it is a way that it produces proteins that the thymus doesn't need itself in order to survive. But there'll be things like, like myosin or something like that you would find in a heart muscle, or there might be something that you'd find in a pancreas. All of this gets expressed in the thymus so that the T cells, when they're undergoing this negative selection, they have access to kind of all of the proteins that would be in your body, even if they're not normally made in the thymus. Somebody asked a question in chat. Let me see. Are there certain characteristics of proteins an APC engulfs that determines which peptide sends to MHC for presentation? In other words, when the protein is chopped up in the APC, could any peptide end up being presented by MHC, or are there certain peptides APC prefers to present due to some property of the peptide? That is a great question, and the answer is yes and no. So in a steady state, if a person is not infected with anything, all of the proteins that are going to be expressed in HLA molecules are going to be self proteins by necessity, because that's the only proteins that are going to be around there, right? That being said, each HLA molecule has a different, what's known as a peptide repertoire. So they have specific, so I showed this pocket a few slides ago, right? Yeah, so this one here. So this pocket, different HLA molecules all have slightly different pockets, which leads them to have different types of peptides that bind in there. And so to answer your question, yes, each individual HLA molecule will have some peptides that it will be able to present really well, and other peptides it can't present at all. And it's actually one of the reasons why we have so many different HLA genes, because if we have lots of variability in the types of peptides that can be presented, that at a population-wide level, there should be an HLA molecule that can present essentially any peptide under the sun. And it also means that at an individual level, we have multiple different genes, so we should have a nice collection of HLA molecules that can present a wide variety of different peptides. But when you look at any one HLA molecule, not each one of them will be able to present every peptide under the sun. They all have different specificities for the types of peptides they can present. So a really great question, and hopefully that answered it. All right, so this actually leads very well into, now I'm going to talk a little bit more about HLA self and non-self. So I just brought up this point, but one of the things about HLA is it has incredibly high diversity. And that diversity comes in a couple of different ways. The first way is that there are 6 different HLA antigens, as they're known, or HLA proteins. So there's the HLA A, C, and B, and those are what is known as the class 1 HLA molecules. And then class 2, there's the DR, DQ, and DP. So in each individual, you have these 6 different antigens, and we have to keep in mind the fact that humans are diploid, so there's 2 copies of every chromosome. So we can have up to 12 or more different HLA molecules. So there's different versions of these HLA molecules, and it's actually up to 18. And I'm not going to get into why that is, but the genetics of this are more complicated, and I might get into that a little bit further. And on top of that, so each person, each one of us here has up to 18 different antigens within us. But then when you look at a population-wide level, there are lots of different versions of each one of these genes. Right, and these versions of a gene are called alleles. So for example, the HLA-A gene has thousands of different alleles, right? Different types of HLA-A genes. And so just to get the nomenclature out of the way here, a type of gene is called an allele, and then each gene itself is called a locus. Locus is one gene, loci is plural for more than one gene. And so when we look at the alleles, all the different versions of those genes, there are currently over 38,000 known HLA alleles. So there's thousands of HLA alleles. So the idea that I'm trying to show here is that there are lots of different HLA alleles out there, lots of different genes. There's just a lot of diversity HLA. And again, the big reason for that, there's a lot of selective pressure during evolution to increase the diversity of HLA genes to make sure that different peptide antigens can be presented to T cells, so that if we're ever infected with something, we will have the ability to present any of those peptides that's never been seen before. Again, a really nice example of that is COVID, right? This was a germ that had never infected humans before 2019. But even though it had never infected humans before, because there are so many different HLA molecules out there, every single person had at least one HLA antigen that was capable of presenting those peptides from this novel virus and inducing an immune response. All right, so finally, now getting to clinical HLA testing. So what does an HLA lab actually do? And there's really 2 main functions of an HLA lab. The first is what's known as immunogenetics. And that's just a fancy way for saying that we're trying to figure out the HLA alleles the patient has. And so this is colloquially known as HLA typing, right? So if you type a person for HLA, you're trying to figure out which specific alleles they have at each of their HLA loci. This is also sometimes known as histocompatibility. The other function of an HLA lab is what's known as transplant immunology. And there we're trying to determine any immune responses a patient might have to foreign HLA molecules. And I'll talk a little bit more about how that works. But the big thing is that HLA labs were really concerned about this self versus non-self axis and how HLA functions with that. All right, so I'm thinking a little bit more about HLA self versus non-self. HLA labs are there to really support the transplantation programs. About 90% of the tests that we do at the HLA lab here at HUP is in support of transplant programs. And so the reason for that is because HLA is one of the biggest ways that your immune system is going to determine if a foreign organ that's put into a patient is going to be seen as self versus non-self. So the idea here is anytime you have a mismatched HLA, that is a potential for your immune system to view it as non-self. So what I'm showing here is this is a heart donor typing up here, and this is the patient down here. So if you look at just the first antigen, HLA-A, you can see that the patient has an A3 antigen, and the donor has an A2 and an A3. Well, this A3 in the donor is going to be seen as self by the patient, but this A2 is going to be seen as non-self. So that is a potential that the patient will view this A2 as a non-self protein and potentially make an immune response to it. We can go through here through all the different antigens. You can see at B, both of these would be seen as non-self. Same for C, same for DR, 13, 15. So there's lots of potential HLA alleles that the patient or the recipient would see as potentially non-self. So, when we think about this, right, especially in solid organ transplantation, both in kidney and in heart, I'm showing here, very often we have lots of non-self HLA molecules when we put an organ in. And so this is going to be, kind of high on the non-self type of axis. And so one of the things that happens during transplant is they also try to reduce the danger safety axis to be more to the safety. That's outside of the scope of the talk right now, but they have different ways of when they put that organ in, trying to make the body accept it as safe, even if it is seen as non-self. All right, so let's see. Some of these can be kind of review, but I've talked about how the function of HLA. So HLA is the human major histocompatibility complex, MHC. So sometimes you will see HLA and MHC kind of used interchangeably. They're really the same thing. And as I talked about, the main function of HLA is an antigen presentation. So this is the same view that I showed before. And again, this is what a T cell would see over here as it's trying to determine if the HLA molecule and the peptide in there is self versus non-self. So when we think about how an immune response to an allograft, to a transplant would happen. So here I'm showing a donor kidney that was placed into a recipient. And so one of the first things that happens when they'll put this organ in, they'll attach all the vasculature, and so they'll attach this new organ to the recipient's blood system. And so one of the first things that happens is antigen-presenting cells, like a dendritic cell that was already in this donor kidney, can come out here and start interacting with the recipient's immune system. And so one of the things that can happen, right, you have this non-self dendritic cell that's going to have a non-self HLA molecule and a non-self peptide being presented in that HLA molecule. And what can happen is a T cell from the recipient can come here and start interacting with this. If this T cell recognizes this, it can start activating an immune response. And this is what's called direct allorecognition, because the recipient T cell is directly recognizing a foreign HLA molecule, a foreign peptide on a foreign dendritic cell. Another thing that can happen is you can have a dendritic cell or an antigen-presenting cell from the recipient come and interact and start getting antigens from the donor, right? So now we have here, this is a self dendritic cell, a self HLA molecule, but it has a foreign peptide being presented in that HLA molecule. And what can happen here is that recipient T cell can also recognize this as foreign because it's recognizing now just this peptide. It's being presented in a self HLA molecule, but it's still a non-self peptide. And this is what's known as indirect allorecognition.
Is there a greater likelihood of one of these happening over the other? In particular, if you have a direct allorecognition, that I assume requires the T cell can interact successfully with the donor's dendritic cell to begin with. You mentioned at one point that's part of the check. Does that make this less likely to happen?
So it's actually more likely to happen. One of the things that happens during negative selection is a lot of the really strong interactions with a self-peptide are kind of removed. And if you have a foreign HLA, it's been estimated that something like 10% of all T cells will react with the foreign HLA molecule, regardless of what peptide is being presented there. And so that is actually a fairly strong way to activate these T cells. However, it's more time limited, right? Because this requires a dendritic cell that came with the donor organ. And those are kind of short-lived cells, right? So this really, the direct allorecognition is thought to happen in the first couple months of after transplant. Whereas this indirect, you're always going to have more and more antigens from this kidney because that kidney should be there for several years. And so indirect allorecognition kind of takes over as the primary way later on after transplant, but the direct allorecognition kind of happens sooner after transplant. All right, so both of these will result in activated T cells that can then go and recognize the allograft and directly injure it, either by killing cells, by activating other cells that will induce inflammation, by activating antibodies, etc. In fact, that gets to antibodies. One of the things that these T cells can do is they can go and provide help to the B cells, and those B cells then will make lots of antibodies, and those antibodies then will directly bind to the kidney allograft and can also mediate graft dysfunction or some sort of rejection of the graft. All right, so that was kind of a background on transplant immunology. I'm going to talk a little bit more in the rest of the talk about how we measure some of these HLA antigens and how we go about typing, etc. So I'm going to start with a tiny bit of history. HLA antigens were first typed by serology. And what that meant is that we had some anti-sera, so some blood products, either from humans, or sometimes we would get them from other animals that had been immunized with human HLA molecules. But we had these serum that had a bunch of antibodies that recognized specific HLA proteins on them. And so what we would do is we would take these serum that had lots of antibodies, we'd incubate them together with a lymphocyte that had these HLA molecules on it. And if any of those antibodies bound to the HLA molecule, we would wash everything else away and these antibodies would remain bound. We would then add exogenous rabbit complement. Complement is a part of the immune system that interacts with antibodies and that actually leads to killing of cells. And so the idea here is if you had these antibodies that were bound to the HLA molecule, they would what's called fix complement, and that complement would actually kill the cell there. And then we would look under the microscope at all these wells, and we would see which wells had live cells, which cells had dead cells. But the example here is, if you had a serum that was called A2, and if that serum reacted with the patient's cells and killed them, then that meant the patient expressed the HLA-A2 antigen, right? And so that was kind of the original way that we had to type HLA molecules. However, we don't do that anymore. What we do instead now is we use molecular methods where we're actually looking directly at the DNA, the genes of HLA, instead of looking at the antigens. And so since we're doing everything by molecular methods, we need to know a little bit about HLA gene complex. So as I think I might have mentioned before, the HLA gene complex is on the short arm of chromosome 6. And if we zoom in here, we can see that these genes are organized into different regions. There's the class 1 region, which has the HLA A, B, and C antigens, and then the class 2 regions, which has the DR, DQ, and DP antigens. As I mentioned before, the HLA gene complex is actually the most polymorphic region in the entire human genome. What I'm showing here are the number of HLA genes that have been characterized and given names by the World Health Organization. And you can see that over the years, we've been finding more and more and more HLA antigens. We're showing class 1 in green here, class 2 in black. And what you can see is starting, about 18 years ago, when many of you were born, there's like 1/10 of the number of genes that we have now. So clearly there are lots of genes out there. We're discovering new ones all the time. Even in the HLA lab here at HUP, we routinely will test a patient and find that they have an HLA allele that no one has ever seen before, hasn't been characterized, and doesn't have a name. And so, this is just something that happens. It's been estimated there are probably something like 3 to 6 million different HLA alleles just on the class 1 side alone. So clearly we've only kind of scratched the surface of the different HLA alleles there. All right, so I've talked about how there's two different classes of HLA molecule, the class 1 and the class 2. The differences are in that class 1, they are encoded by a single locus, a single gene, either the HLA A, the B, or the C gene. And so here I'm showing a cartoon of how a class 1 HLA molecule would look. This is the cell membrane here. This is intracellular. This is extracellular on the outside of the cell. And you can see that the biggest region of the HLA gene is on the outside of the cell. And this peptide binding cleft where that peptide would bind is furthest away. All HLA molecules are what is known as heterodimers, meaning that two proteins form the entire functional protein. And so for class 1, the majority of this is encoded by the HLA gene, either the A, the B, or the C. And the last little bit of it is encoded by what's the protein is called the beta-2 microglobulin. This is a protein that's encoded by a different gene completely outside of the HLA region on a different chromosome even, but it's just something that helps the HLA molecule form. Class 1 HLA, they are expressed on all nucleated cells. So basically every single cell in the human body has a class 1 HLA on it except for red blood cells. Class 2, on the other hand, is a little bit different because now you have two separate genes from the HLA region that form that heterodimer, right? So for example here, this alpha chain of the protein would be encoded, for example, by a DQA1 gene. And this beta chain would be encoded by a DQB1 gene, right? So you have two different genes that form this functional heterodimer on the cell surface. And because of that, there's also both the alpha chain and the beta chain have variability. So you can have different combinations, all right? And because if we just give DQ as an example, right? You have the DQA1 gene that you might have two different alleles, right? Because you have one on one chromosome, one on another chromosome. DQB1, you have two as well. Those can all form different combinations of those four genes. So you can have up to 4 different DQ genes just from the 2 genes that you have, which is different than class 1, where you can only have up to 2 for each locus. The other big thing, difference for class 2 is that they're only expressed on antigen-presenting cells. So these are going to be things like B cells, dendritic cells, and macrophages. All right, but again, I talked about how when we're doing typing, we're looking at the genes, we're using molecular methods, we're looking at the DNA. So we need to know a little bit about the genetic structure of HLA genes. So I'm showing here an example of the HLA-A gene. And so just as a review of molecular biology, there are different parts of a gene. We have the untranslated region, both on the 5' and the 3'. And so these are regions of the gene that are transcribed to messenger RNA, but they are not translated to a protein. In the middle here, we have these introns. And so these are sequences that are spliced out of the mRNA before they're translated to a protein. And then finally, we have the exons, and these are the actual parts of the gene that get translated to a protein. So this would be what would be known as the protein coding regions of the gene. And this is important because those exons correspond pretty well to the big regions of the HLA molecules. So in class 1, exon 2 and exon 3 encodes for this peptide binding cleft. And again, that's the part that presents the peptide to a T cell. And so this is really the most important part of an HLA molecule. When we're talking about some of the amino acid positions that we have seen that fibers has discovered, for example, they are often at this part here. And that's why we get so excited, because they're parts that are either interacting directly with the T cell or interacting with the peptide and leading to different types of peptides that can be presented. And that really makes us think that that's why the T cell response is more important to this as opposed to the B cell response, because a B cell could bind to any region, but this is really the part of the HLA molecule that interacts with a T cell. And so that's why we get really excited when we see all these amino acid positions at these peptide binding clefts. Class 2, it's similar, where exon 2 are the part that encodes for these peptide binding clefts. So I talked a little bit about historically, we typed everything by serologic methods, but now we're doing everything by molecular methods. And we have several different assays or technologies that we use. They're given different names like RT-PCR, SSO, next generation sequencing, but they all use these fancy machines these days. And now that we're dealing with HLA typing, we have to know a little bit about the nomenclature of HLA. And so I'm showing here a name for an HLA allele. And you can see that there's a lot going on here. It's fairly complicated. But I will break it down. So the first part, HLA-A, that's just telling you the gene that you're looking at, right? So HLA-A is going to be one locus, HLA-DRB1 will be another locus, etc. This separator, this asterisk, whenever you see this asterisk, you know that you're looking at molecular nomenclature compared to the older serologic nomenclature, which does not have that asterisk there. And then after this asterisk, this is where you actually have the gene name. And you can see that there's 4 different fields, as they're called, that are separated by these colons. And this first field is what's known, encodes for the allele group. I like to teach that this is like being able to type someone to their last name. So if you were to type me to the first field, you could see that I'm part of the Brown family, but you wouldn't be able to tell me apart from my sister or my parents, etc. The second field there is if you are able to type to that second field, now you know the specific HLA protein. So if you were to type me to the second field, you know that I'm Nick Brown, I'm not anyone else in my family there. And so for the most part, clinically, these first 2 fields are all that we ever report. And certainly in our larger project, these first 2 fields are all that we're really interested in. There's 2 more fields. This third field here is used to show any synonymous DNA substitution with codon region. The idea there is that the DNA sequence is degenerate, meaning there are different DNA sequences that encode for the same protein sequence. And a difference here is just showing that there's a different DNA sequence, but it still encodes for exactly the same protein. And then finally, this last field is used to show any difference in a non-coding region, which shouldn't, but sometimes does affect expression. And then lastly, you can have some suffixes that are added there, most important of which is the N, which stands for null allele. And that's anytime we have determined an HLA allele that has a mutation somewhere in it that can't produce a functional protein, it will get an N there. One of the other things that happens is because HLA is so polymorphic, because there are so many different HLA alleles in the human population, it's not always possible to know the exact HLA allele that a patient has or a donor has, for example. Sometimes all we can do is know what's called like low resolution, for example. And the idea here is if we are to type somebody to low resolution, that's essentially equivalent to knowing their first field. And again, it would be like typing me low resolution would mean that you could type me only to my last name, but you couldn't figure out what my first name is. And this is also usually synonymous with serologic nomenclature, right? So this is what's showing here. This A2 is in that older serologic nomenclature, and the A*02 would be in the newer molecular nomenclature. High resolution, on the other hand, would mean that you were able to determine the exact protein that is going to be made by the individual. And high resolution is what we're trying to get with the data that we're getting out of the 10 different labs. We're trying to get their high resolution type so that we don't have to use the older low resolution type, but we'll actually know the specific protein that we're getting out of these labs. And in fact, you can even have a resolution that's higher than high, and that would be considered allelic resolution. And that is if you know the entire DNA sequence of the entire gene, you could tell all 4 fields from it. So, I've talked about how we currently use molecular nomenclature, but I talked about how we used to do things historically. And there's a reason for that is even though we don't use those tests anymore, we still have to report lots of things to UNOS, to the federal government who helps us identify organs for deceased donor transplantation. We have to report out everything in serologic nomenclature. Even though we haven't been doing those tests for decades now, they still require us to report everything in the older nomenclature. So we have to have some way of translating between the 2 of them. And so for the most part, it's pretty straightforward, right? So we might type a patient and find that they have an A*0201. And usually what we do is we take that first field and that gets translated to A2, for example. This DRB1 1454 would be a DR14 because that's that first field there. And for the most part, that's pretty straightforward. However, there are some things that are what I like to say is lost in translation. So for example, this DQ7 in the older serologic nomenclature is actually encoded by the DQB103:01 gene. And you can see that 7 is not at all evident anywhere in this molecular nomenclature. And that's for kind of historical reasons I won't go into, but there are a bunch of these alleles that are kind of the exception. So we have to be able to kind of tell between the 2 of them and translate between serologic and otherwise. Now, when we're thinking about how this all comes into play for kidney transplantation, a big part of what is going to determine the outcomes for a kidney transplantation is how good the HLA matching is. And so the way that would work, as I'm showing here an example of a recipient typing at the A, B, and DR, and a donor typing at the same one. If we look at this donor here, we can calculate mismatch scores for each of the locus, right? So if we look at A here, we can see the donor has A1 and A2, which is the same as the recipient. So the recipient is not going to see anything as foreign there. So a mismatch at this locus is going to be 0. If we go over to the B locus, we can see recipient has 8 and 44, but the donor has 8 and 62. So this 62 is going to be seen as foreign by the recipient. So this is going to come out to a 1 mismatch at this locus. The same thing for DR. So the total mismatches for this donor is going to be 2 mismatches. And this is important because here I'm showing a very long longitudinal study that's been going on for many years. They're looking at kidney transplants from 1990 to 2022. And you can see that as you get more and more mismatches, you have worse and worse outcomes, right? So you have 0 mismatches in black here, 1 mismatch in red, 2 in blue, etc. So the more cumulative mismatches you get, the worse the outcome is going to be. Another way of thinking of that is the more mismatches you have, the shorter amount of time that kidney is going to function in a person before it's rejected. And so again, the way this is done is with that older serologic resolution. However, we're doing things these days with molecular nomenclature where we have more information on the exact protein that's being made there. So what we're trying to do these days is we're trying to figure out better matching algorithms instead of looking at the entire antigen, just saying, all right, this is an A2, this is an A68, they're different from each other. What we're trying to do is say, all right, well, this A2, this is encoded by this polypeptide chain, right? Where we can figure out each amino acid that's going through here. I'm showing just an example of a protein, and you can see the different amino acids that are made in there. And we're saying, all right, can we, instead of looking at the entire antigen, start looking at these individual amino acids and find if some of those amino acids are more important to be matched than other amino acids? So an example of how that would work is here I'm showing the amino acid sequences. What I'm trying to show here is each color would be a different amino acid. And so for a recipient, you can see they have 2 different proteins that are made because we have 2 chromosomes, 2 sets of genes. And if we look at this one position, let's say this is DRB1, the 13th amino acid position. Well, the recipient here has an arginine and a serine. The donor has an arginine and a phenylalanine. And so what this means is that this phenylalanine, this F, is going to be seen as a mismatch at this particular position. And so at DRB13, there's going to be 1 mismatch. And so Ryan and Keith, this is what the Fibers is doing. It's looking at these individual amino acid positions and figuring out when there is going to be a mismatch there. I want to spend a couple more minutes talking a little bit more about the actual data. A lot of the people that are on here are going to be actually working with the primary data, and I wanted to finish with how some of the data looks here. So one of the things that we do is we have a lot of assays that result in what's known as ambiguities, right? So you type a patient and you can't tell for sure which allele the patient has. Instead, what you do is you take those 38,000 known alleles and you restrict it to a much smaller set of potential alleles that a patient has. So here I'm showing an example from an assay that's called the RT-PCR assay. This is used widely in solid organ transplantation. And you can see here that we've typed this patient or a donor, and it says, all right, well, it has the A02 group, but these are all the potential alleles that the patient could have, right? It could be an A0201, 0101, or an A0201, 04, or an A0204. All of these are ambiguities, meaning all of these are completely consistent with the results we got, and we don't know for sure which one the patient actually has. I wanted to give an example and talk about imputation, because one of the things that we do in this larger project is we take the serologic resolution and we try to impute which potential allele that they might have. So I wanted to give an example here of A24. Let's say that we got from this governmental organization that a donor was typed as A24. Well, if we go in online to this website that catalogs all of the different alleles, and if I just put in A24, you can see that there are 994 different alleles that are part of this A24 group. Right. So if we just get an example of A24, well, we don't know which one of those 994 different alleles it could potentially be. But one of the things that we can do is we can use haplotype frequencies. But the whole idea of a haplotype is a set of genes that are all encoded on the same chromosome and tend to be passed down from parents to children as a block. And I don't know why this picture looks so terrible, but what I'm trying to show here is this is some nice data we have on haplotype frequencies, meaning, all right, so this potential haplotype here would have an A24 and a C, oh, I can't see what that is. I don't know why the picture looks so bad. Let's say it's a CO303 and a B0702. What we can do is we can say, all right, well, these sets of genes tend to be inherited together. So what we can do is we can take information from a different locus, right? So for example, we say, all right, we want to figure out which A24 allele the patient has. Well, we also know that that patient was typed as a B7. So if I take this same thing and I now filter it for any haplotype that has a B7 in it, you can see it restricts that down to only 5 potential haplotypes there. And if we look at all those haplotypes, you can see that A2402 is in all of them. So this is a really good example of how we can use imputation. We can use data from 2 different loci to get a much better idea of the potential allele a patient has. And so this is one of the things that Dr. Greger does. He specializes in imputation here. And I just kind of wanted to give a really quick background on how that worked. But I'm over time. This is all the slides I had. And I kind of wanted to stop here and open it up for any questions. So Praneel, you have your hand up.
Thank you for the information, by the way. That was great. In the, I think it was the first fibers paper, there was a thing that said that to actually get this imputation with haplotype frequencies, I think it talked about multiple imputations. So could you explain like what that process is and how it's...
Yep, that's a great question. So the way multiple imputation works, so there's these 5 potential haplotypes, and you can see that there's a different frequencies of each one of them, right? So this one is at 0.9%, this one's at 0.1%, etc. The way multiple imputations would work is if we have determined based on the data we have that only these 5 haplotypes are possible, the way multiple imputation would work is it would take a random number between 0 and 1, and each one of these haplotypes would get a different piece of the potential pie, right? This one is the most frequent, so it would get the biggest piece of the pie. The next one would get a smaller piece, etc. Using that random number, wherever it would fall on these 5 haplotypes, it would take that haplotype and put it in there. And then multiple imputation means that we would do that 10 different times. So we would take the same base data, we would do 10 random imputations, and each one of those is going to have a slightly different set of possible alleles based on their allele frequency. But that will come out with 10 different alleles, and then we can use that to get a better idea of it's a way to kind of capture the remaining ambiguity in there without just picking the most common one, for example. So does RT-PCR not provide the exact genetic sequence of the sample's HLA gene to get the real allele a patient has? That is correct. It does not. So I didn't have time to go into that. I have whole talks about how the individual assays work, but RT-PCR, for example, so the entire HLA gene is roughly 3,000 to 10,000 nucleotides or more. The way some of these assays work is they only can look at maybe 100 different positions at a time. So clearly, if you have a gene that is 3,000 nucleotides long, but you can only look at 100, you're not going to be able to look at the entire gene. So what they do is the companies that make these, they try to design primers or probes that will look at the most frequently changed positions or the most importantly changed positions. And so you'll come out with a much smaller set of possibilities, but you don't know for sure which ones you have. Other assays like the NGS, which stands for Next Generation Sequencing, that is an assay that sequences the entire gene. And so when we use those assays, we can get what's known as high resolution. We know the exact allele that a patient has. And one of the things that we're doing in this larger project is we are trying to get that data out of the individual HLA labs, because this is something that many HLA labs, including HUP, has, but it doesn't make it to the governmental database. It stays within the HLA labs. So one of the things we're doing is trying to go to each one of those labs and get out that higher resolution data, because what that will mean is that we won't have to do this imputation. We'll know for sure which exact allele the patient has for at least more of the transplants than we do now. And that will make our error bar smaller, and it will make it much more likely for us to know which amino acids are actually important. Yeah,
just to further connect everything that Nick has said to the projects we're actively working on. So with respect to fibers, our first paper and all the papers that deal with the real world analysis so far have relied on the imputed amino acid information that Dr. Graggart had prepared for us. And we've been developing fibers just using simulated data and this imputed data thus far. And so we're doing as much as we can to develop the methodologies, demonstrate that they're effective in preparation for this exciting new wave of data that Nick is leading, the gathering of, that will ultimately be the target of applying all these methods we've developed once we've collected all that high resolution data.
Absolutely. Very nice.
Any other questions for Nick?
I'm sorry. It looks like we have one more question. Praneel?
So previously, we were using that multiple imputation strategy. When we get the allelic super high resolution data from like labs like Baylor and so on, is the idea that we're going to apply fibers directly on that data instead?
Yes.
Okay.
Right. Just to be a little bit clear, we are going to get a lot of high resolution typing data, but not all of the transplants that will have high resolution on. But even so, I've been making sure that we get intermediate resolution, which is still better than what we have. And so if we had, ambiguity this big, even with that intermediate resolution, the ambiguity is going to get much smaller. And then when we have the super high res where there's no ambiguity, we're still going to have a very nice big data set with much smaller levels of ambiguity attached to it. But we will still be able to plug it into fibers, and hopefully it will tell us if some of the amino acid positions that hasn't told us before, because there's simply too much ambiguity at those positions, it's never going to be able to tell us that those ones are important. We're hoping that it'll be able to tell us that those ones are important too. All right, well, if there's no other questions, then have a great day, everyone. I really appreciate all the great questions that you guys came up with. So awesome.
Thanks again, Nick. Great.
Thank you.
Thank you.
Thank you.
so much.
Everyone.
Have a good day. Bye.